Hi guys, welcome to our third lecture. This is biochemistry. Now, in the realm of biochemistry, what we're going to be dealing with here are two separate types of molecules. These are the inorganic molecules and the organic molecules, and this is very important for us. Now, the difference here is as follows. Inorganic molecules tend to be very small, and they tend not to contain carbon, but being very small is the key point. For example, carbon dioxide, CO2, two oxygens, carbon. This is a very, very small molecule, and it would be considered inorganic, even though it contains carbon. Oxygen, water, so like H2O, that would be a inorganic molecule. Very small, salts, acids, bases, water. Very small, typically non-carbon containing. Now, by comparison, there are organic molecules. And organic molecules will be orders of magnitude larger. For instance, if water is 3 atoms, like a general organic molecule can be like 300 atoms. They can be very large indeed. And organic molecules will always contain high percentages of carbon. Okay, carbon forms a very stable backbone for our very large organic molecules. So our organic molecules are proteins, carbohydrates, lipids, nucleic acids. Okay, so let's go through and talk about some of these. I believe we might have mentioned this before, but I'll mention it again. Water is the most important inorganic molecule that we're going to come across here. It's so important because it makes up such a large percentage of our bodies, making up a huge percentage of our cells. It's very, very abundant and is a real foundational point for life to exist is water. And the reason water is so important is because it has a high heat capacity. When we say that water has a high heat capacity, that means it has an ability to absorb and release a lot of heat before it actually changes temperature. And because you are mostly water, that means that you have a high heat capacity. Again, the concept is that you can walk out on a really hot day and not die of a heat stroke immediately because your body takes a lot of heat gain to actually change temperature. Or alternatively, you can take the trash out on a freezing cold day and not die from heat loss because it takes a lot of energy loss to change your core temperature. Okay, you have a high heat capacity, which means, key point, you are thermally stable because you're made up mostly water. Water has a high heat of vaporization. What this means is it takes a huge amount of energy to convert water from a liquid to a gas, as an example. And what that means for you is, because you're mostly water, that your evaporative cooling mechanisms are very, very good at what they do. Okay. You have a really nice ability to evaporatively cool, so when you sweat, it liberates copious quantities of energy from your system. And let's see, water is highly reactive. And what it does is either hydrolysis reactions or what are called dehydration synthesis reactions. Now, a hydrolysis reaction, hydrolysis. Lysis means to break up, and hydro, like if you hydrate, means to add water. So we're adding water and breaking up molecules. Okay. All of your digestive reactions are hydrolysis reactions. You have enzymes that will break up other molecules by adding water to them. And alternatively, in your tissues, when you have to heal or grow larger or do any of the maintenance that your body requires and you have to build something, so intercellularly, you have to build something, all of those reactions are considered dehydration synthesis reactions. Dehydration means to take water away, and synthesis means to build. So this is to build something by dehydrating it. All of the mechanisms in your body that allow you to build things are dehydration synthesis reactions. And water is indeed a polar solvent. The general idea here is that water is very good at breaking up most molecules, especially ionic ones. But let's just not worry so much about that at this stage. Alright, pH. So acids and bases are where we're going here. But before we can really talk about what an acid is and what a base is, being that those are very important inorganic molecules, we really need to talk about the pH scale first. Alright, the pH scale is how we measure the amount of acidity or basicity. You will also hear me use the term alkalinity when I'm talking about things that are basic. So this scale runs from 0 to 14. The more towards 0 you go, the more acidic things are, and the more towards 14 you go, the more basic things are. And all acids and bases will break up, they'll dissociate in water, and when they dissociate, if they're acidic, they release free hydrogens, H plus free hydrogens, and if they are basic, they release hydroxyl groups or OH groups. Yeah, that's good enough. So 7 is neutral. Anything towards 0 is acidic. 7 is neutral. Anything towards 14 is basic or alkaline. And worthy of mention here is that this scale is logarithmic. And what that means is that each number is very, very, very different from adjacent numbers. All right. A log scale means it's on a base of 10. So a pH of 3 would be 10 times more acidic than a pH of 4. Okay? A pH of 4 is 10 times more acidic than a pH of 5. So each of these numbers is actually incredibly different from one another. Very, very different indeed. So acids are proton donors. They release hydrogen ions when placed in solution. You have a lot of acids that are very important for you, like hydrochloric acid, which is found in your stomach. There are a lot of acidic byproducts of your general metabolism. Uric acid as an example here. And then, of course, there are bases. Bases are proton acceptors. That's a short way of saying they release hydroxyl groups when placed in solution. And you have a buffering system in your blood that keeps your blood's pH a little bit on the basic side. So bases are certainly very important for us as well. So both acids and bases are used by the body for general function. Yeah, human blood, normally about 7.4, slightly basic. I tell you, I'm not terribly concerned with this. Let's just keep rolling. Organic molecules in the carbon atom. Okay. The point here is that carbon-backed molecules tend to be very strong, and that's where we're headed. Okay, we're going to talk about big organic molecules. So how is it that big organic molecules can exist? Well, that's because bonds with carbon, nitrogen, or carbon, bonds with other carbon atoms, nitrogen, hydrogen, oxygen, what have you, in a very stable way. Carbon bonds tend to be very, very stable, and carbon-to-carbon bonds are very stable indeed, and allows for big old chains of hydrocarbons to be formed. There can be double bonds and triple bonds, big ring structures. Carbon is just very effective at forming large molecules. So let's talk about these large molecules, of which there are 4. These are proteins, carbohydrates, lipids, and nucleic acids. First, we need to discuss monomers and polymers. That's where we're going to be going with this. Now, these big organic molecules, they all have building blocks from which they are made, unique to each type of molecule. Proteins have their own unique building block. Nucleic acids have their own unique building blocks. And what we're going to do here is we're going to string these building blocks together to make big functional units, like starch. Okay? Starch is a huge molecule of many, many glucose subunits. These glucose molecules will get together and chain link together to form a big old starch molecule. A single starch molecule is made up of lots of glucose. The way that we would phrase this is we would say that starch is a polymer. All right? A polymer is a big long chain of many glucose monomers. Glucose would be a building block, a monomer. Starch would be a polymer, a group of monomers covalently bonded to one another. Now, how do we do this? It's actually very simple. We take a couple of monomers and we can assemble a polymer from this via dehydration synthesis. Okay, this is very obviously dehydration synthesis because you can see water leaving. We're actually pulling a hydrogen and an oxygen and a hydrogen off, and that leaves as water. 2 monomers are joined together covalently to form a polymer that is dehydration synthesis. Dehydration synthesis, because we're making something by removing the water from it. By comparison, your digestive processes, if we have a polymer and we have to break that down into its monomeric subunits, for example, we can't absorb large molecules in our intestinal tract. We have to break those down into their monomers. If we have a big polymer, our digestive process is going to break that down via hydrolysis. It adds water into the system and breaks it down into its monomeric subunits. Okay, so hydrolysis is how we break things down. Dehydration synthesis is how we build things up. And that takes us to the sugars, carbohydrates. All right, carbohydrates are indeed sugars, and in the carbohydrates there are several what we call monosaccharides that are the building blocks from which the large polymers are made. These monomeric subunits, if you will, these monosaccharides are glucose, fructose, galactose, deoxyribose, and ribose for this conversation. The three main ones, obviously, being glucose, fructose, and galactose. Deoxyribose and ribose are found in the nucleic acid realm. Deoxyribose for DNA, ribose for RNA. Yeah, let's talk about these. These are all plant-based. For example, there are several ways that we can put these together. For instance, here's glucose, here's fructose. If we were to covalently bond these, that would be taking a monomer and making a polymer from this. That would be dehydration synthesis, and that would make sucrose. Sucrose is good old-fashioned white table sugar that you put in your coffee. Okay, just the standard stuff you buy. You go buy sweeten it with Domino. You're buying sucrose. Your body cannot absorb sucrose directly, so what you do is you use digestive enzymes to break sucrose down into glucose and fructose, which you can absorb and use. When you break that down, you break it down via digestive processes based off of hydrolysis. All right, hydrolysis. And we can play this game all day long. Like, for instance, two bound glucose molecules is maltose. Maltose is what we use as a sugar in brewing. You may have heard of malted barley or malt liquor. So this is a brewing sugar. And alternatively, there is lactose. Lactose is a bound product of galactose and glucose. And this is the sugar that we as mammals feed our young in order to help them grow larger. Perfect. And then, of course, there is glycogen. So glycogen is the only big polysaccharide that we as humans are capable of making, most mammals, in fact. We make glycogen in our liver as a storage form of sugar. So let me explain what happens. You go and you eat, and in that meal you just have a steak. So you have very little sugar coming in. That small amount of sugar that you do take into your system flows around your system and is used by your cells all as well in the world. But what if you go and eat a dozen donuts? Okay, you go eat a dozen donuts. Your blood sugar levels are going to start going up real fast, and you're going to start cranking out insulin from your pancreas to help you deal with this. And what the insulin does, one of its jobs, is to tell your liver, to begin to store glucose. So how do you store glucose in the liver? You take a bunch of glucose molecules and you bind them to one another so that they become immobile, and that is a glycogen molecule. Glycogen molecules are just big old bound up subunits of glucose. We do this so that the glucose molecules are no longer mobile. So binding them up as glycogen prevents them from being able to move around. They're basically stuck in the liver tissue as a sugar in this fashion. And yeah, man, that's how we do it. So glycogen is a very important energy storage molecule for us. We find it sometimes in the muscles, but oftentimes in the liver. In human tissues. Yeah, I like it. Big molecule. And if you recall from diffusion, big molecules don't move very quickly. Now let me say this too. After you've eaten, if you're just sitting around and it's been like, 4 or 5, 6 hours since you've eaten, your body has the ability to release a different hormone called glucagon, and this will actually tell your liver to start breaking down a little bit of sugar and mobilizing it into your bloodstream. The idea is if your blood sugar levels rise too high, you release insulin, that brings them down. If your blood sugar levels begin to drop too low, you release glucagon, that brings it back up. All by either breaking down or building up glycogen. Think of glycogen as a bank in which you store your sugars. Let's see, there are other polysaccharides in the world around us that are very important. Starch is a big bound-up form of glucose that's found in potato tubers, as an example. This is a good old-fashioned plant sugar that we are capable of utilizing. We have enzymes that let us break down starch. Here is glycogen, which we just talked about, which is found in our systems, mammalian systems. And then obviously there is cellulose. Cellulose is pretty cool. Cellulose we are not capable of breaking down. This is plant fiber. When somebody says you need more fiber in your diet, or talking about plant fiber, or that a salad has a lot of fiber, what they're saying is the salad has a lot of cellulose in it. Cellulose is just a bound-up form of glucose, but the way in which it's chained together, we do not have an enzyme that allows us to break this down. So cellulose is a very strong structural component of plants in most cases. And there are other forms of this bound-up glucose as well. Peptidoglycan is found in bacterial cell walls, as an example. Chitin is found in crustacean exoskeletons. It gives it its hardness. All of these are polysaccharides in the grand scheme. All right, the lipids. So the general lipid, well, hang on, let me say this. When we say lipids, we're talking about fats and oils, phospholipids, steroids, what have you. And the general lipid that we think about are the triglycerides. Okay, triglycerides are fats and oils dependent upon their general structure. They're built of a glycerol backbone with 1, 2, 3 fatty acid chains. And generally speaking, there are saturated fats, which are saturated triglycerides. There are unsaturated fats, which are unsaturated triglycerides. Now, what the heck does that mean? What we have here for a saturated fat is that all of the carbons in these fatty acid chains, okay, all these carbons, they are saturated with hydrogen. There's a hydrogen bound to all of these carbon options here. Whereas in unsaturated fat, you'll find one or more double bonds in place. And you got to think about this kind of like Tetris, all right? When you play Tetris, the straight pieces or the boxy pieces are really easy to line up, and you can stack them anywhere the heck you want. But the parts that are kind of curvy, you got to put them in unique places to make them work, and it's just kind of hard to stack them. Because saturated fats have straight tails, they tend to align quite easily at room temperature, and they form solids. So saturated fats tend to be of animal origin, and they are solids at room temperature. You think about your bacon grease or your lard or your butter, all of this, which is solid at room temperature, would be of animal origin, and that would be a saturated fat. Whereas unsaturated fats tend to be of plant origin. These are like olive oil and canola oil and what have you, general vegetable oil. And that's because their kinked tails prevent them from stacking up at room temperature, so they stay more liquid. You can take these and put them in a refrigerator and they'll turn solid. But at room temperature, they are liquids. These are unsaturated plant origin fats. And worthy of our mention here, I suppose, are omega-3s and trans fats. Right? Omega-3s are considered heart-healthy triglycerides. When you have a high amount of omega-3 fatty acids in your system, this tends to decrease your risk of heart disease. And then by comparison, trans fats are incredibly dangerous, and they're built off of this system. What a trans fat is a partially hydrogenated triglyceride chain set. Okay, I remember years ago I lost a hash brown from a fast food place in the floor of my Prius, and we didn't find it for like 8 months. And when we finally did find it, it looked just like the hash brown that fell on the floor. It looked no different. And the reason for this is quite simple. It had been cooked in trans fats. Okay, so because it was cooked in trans fats, it did not denature, did not break down. Trans fats are where we take a normal bent fatty acid chain like this with a double bond, and in a laboratory setting, we partially hydrogenate it, which twists the molecule a little bit. It just twists this over, and it makes the tail look almost straight in its molecular format. Okay, it makes the tail act almost straight, but not quite straight. The idea is this is a molecule which is not seen in nature, and because it's not seen in nature, there is really nothing that can break it down. You drop this on the side of the road, man, it can stay there for an eternity. You go and pause this video and look up McDonald's food that's been left in the closet for a decade. It comes out looking exactly the way it went in because it's built with trans fats in its cooking mechanisms, and they just don't break down, okay? By comparison, in your system, when you consume high amounts of trans fats, like if you're eating a lot of McDonald's hamburgers, what will happen is these trans fats will flow in your system, and you don't have enzymes that can break them down either, so they just kind of stay in your blood flow and end up causing heart disease. Okay, they tend to form arterial plaques because the longer the residence time of a fat in your system, the higher the risk of arterial plaque buildup. So trans fats are, do I have another slide? Yeah. According to the FDA, trans fats are not generally recognized as safe, which is a fancy way of saying to you that you should probably stay the hell away from trans fats. All right, then there's phospholipids. So phospholipids are, they look like triglycerides. What we have is a glycerol backbone, 2 fatty acid chains here, but then instead of the 3rd fatty acid chain for a triglyceride, what we have is a phosphate group. And what's fascinating about this phospholipid is that this phosphate group displays polarity. So this is polar covalently bound, whereas every other oil is non-polar covalently bound. What this means is that the polar end of this is capable of binding to water, whereas the nonpolar end of this is capable of binding to oils. And as a result of that, we tend to use phospholipid-based molecules when we're building soaps and things. So if we need to clean grease off a pan or what have you, and I'll show you a video of that, what will happen is we'll use molecules like this so that the polar group can bind to the water and the nonpolar group can bind to the oil, like baking grease on a pan as an example, and you can wash the grease away. You'll see the water turn like a milky white color, and that's the grease dissolving into the water. Not only that, but these phospholipids also have a tendency to align themselves. So if you get two phospholipids together, they will align with head to head and tail to tail like this. And if you get a bunch of them together, they'll all just line right up. And that's why we use certain soap molecules that are based off of phospholipids to build like bubbles you'd blow for your kids, you know, and I'll show you a video of that too. What's happening there is these molecules, they tend to form these lines, and when you blow through them, they'll snap off and form little bubbles of basically phospholipids. And you use this in your system as well. So your cell membranes are based off of phospholipids. Your cell membranes theoretically are a phospholipid bilayer. And what will happen is this is two layers of phospholipids, hydrophilic on the outer surface and the inner surface, and hydrophobic in the core. These are hydrophobic oily tails that don't let polar molecules through, and then these are polar heads that don't let nonpolar molecules through. The gist of this is that phospholipid bilayers like this for your cell membranes are what we call selectively permeable. This makes your cell membranes selectively permeable. In other words, they don't just let everything in. And that's what I'm kind of showing you here. You can see our bubbles here a little bit. You can see this fluid mosaic of our cell membranes. Yeah, man, this is all good stuff. Alright, then there are steroids. Steroids are actually built from cholesterol molecules. So here's a good old-fashioned cholesterol molecule. These are very important as precursors to all of our basic steroid hormones. And when I say steroids, I don't just mean, you know, professional athletes doing what they do. Steroid hormones also include our sex hormones like estrogen and testosterone, and also vitamin D. And these are all very, very important for us. Before I get into this, let me jump down here. Cholesterol also matters in our cell membranes because it helps to keep our cell membranes fluid. If our cell membranes get too hot or too cold, the cholesterol helps to maintain stability and fluidity within our cell membranes, which is very important. All right, vitamin D. Let me make sure I'm not going to jump ahead here. Good. All right, vitamin D is a steroid hormone made from cholesterol. It's a lipid that will allow us to absorb calcium from our gut content. So when we eat food, we have to have vitamin D for the absorption of calcium. Whereas testosterone and estrogens, also based off of cholesterol molecules, give rise to our male and female secondary sexual characteristics, without which we would not be capable of breeding, and that would mean collapse of society as we know it. So these are both very, very important for us. These molecules, like these cholesterol molecules, even if you didn't consume food that contained cholesterol, your body has the capacity to manufacture new cholesterol to keep the system functioning. So this is very important. So cholesterol is not the enemy, as it were, even though a lot of your dietary knowledge probably would say otherwise. And I like to put these charts onto these slides. I oftentimes don't make you buy books, and when I do, I don't know if everybody's reading it. So what I try to do is I include these charts from the text that will help you to sort of put who's who together to figure out where these molecules fall. So this is for you to read and help you study. All right, let's jump right into the organic molecules, the proteins. Okay, so proteins are also organic molecules. And proteins are just super, super important for your general daily function. Proteins are things like enzymes and the contractile proteins of your skeletal muscles that allow you to move. Oxygen carriers in your blood like hemoglobin. I've said this to you before, but when I see you, your hair, your skin, your nails, all of that's all protein. All your muscles, it's all protein. So proteins are very important. Proteins are made of smaller building blocks called amino acids, of which there's about 20 different types. These amino acids bound together form up proteins. Now, I need to make a point here, and you're going to hear this. This is very tedious, and I hate tedious things, but you're going to be hearing about it in the future, I predict, so we might as well discuss it now, and that is the concept of the peptide bond. Now, proteins are made of amino acids which are covalently bonded together. They're covalently bonded amino acids, and that makes up proteins. But we don't call them covalent bonds when we're dealing with proteins. We call them peptide bonds. Anytime you hear the term peptide, you need to be thinking about protein. Okay, these are proteins. Peptide bonds are just the name we assign to the covalent bonds which form between amino acids which are making up protein. Yeah. Yeah, I tell you what, I'm pretty happy with that. That'll do. So peptide bonds. Now, proteins have a lot of different structural builds, if you will. So when you first start making a polypeptide, as it were, you form up this primary strand of a protein. These are basically just peptide bonded amino acids, a whole bunch of amino acids, amino acid, amino acid, amino acid, amino acid, amino acid, all peptide bonded together to form a long string. This is not a functioning protein. What we will then do is we will allow this covalently bonded backbone, peptide bonded backbone, to form up either by sort of making a sheet like this, which would be considered a beta-pleated sheet, or by spiraling together and anchoring to itself, and that would be called a alpha helix. Now, pay attention, this is important. The structure here is peptide bonded. This is covalent. But all the unique shapes that proteins take on, this is so important. Please listen. All of the unique shapes that these proteins take on, this is due to hydrogen bonding. If you recall, hydrogen bonding being weak attractions between polar covalently bonded molecules, where there is polarity, you can have hydrogen bonds. That's what all this is. These little dashed lines. I'm going to ask you this on your test, man. You better be familiar. All these little dashed lines, those are hydrogen bonds. And hydrogen bonds are weak attractions, man. They're weak attractions. The side comment here being that if you heat up a protein, you can denature these hydrogen bonds and yield the enzymes to become non-functional. Denaturing means to break up the hydrogen bonds. And you've seen this happen before. If you've ever grabbed something that's too hot and you've burned yourself and you can see the burn mark on your hand, you busted up the hydrogen bonds in the proteins of your skin. Okay? If you've ever taken a steak from Publix, or if you're smart, somewhere else it doesn't cost $25 an ounce. Anyway, take a steak, it looks red, and you throw it on the grill and it turns darker coloration. What's happening is you're breaking up the myoglobin, which is a protein in that steak, okay, in that meat, in that muscle tissue. Alright, so we have primary structures, we have secondary structures like beta-pleated sheets and alpha helices. These can then further twist upon themselves and form a tertiary structure of a single polypeptide chain, and then alternatively this can form a quaternary structure where you have two or more polypeptide chains coming together and hydrogen bonding to form a unique structure like this, which is hemoglobin. This is the oxygen carrier in your bloodstream. It is a quaternary protein structure. Yeah. So general functions of proteins like metabolism. Most enzymes are proteins that act as catalysts to accelerate chemical reactions within cells. That's a fact. So all of your enzymes, these are protein-based. Support, like keratin in your skin and collagen. Keratin and collagen are indeed protein-based, and they give rise to the structural rigidity, if you will, of your tissues. Their ability to restrain tensional or torsional stretch. Transport, like hemoglobin as an example, carrying oxygen in your systems. Your antibodies are protein-based. Hormones, yeah, man. Motion, actin and myosin in your muscle leads to flexion, and that's all protein. This is really important. This looks like a nice multiple choice set for you. You need to be friends with it. Check my time. Alright, good. Then there are, of course, the nucleic acids. So in the realm of nucleic acids, we need to talk about the building blocks, and these are the nucleotide base pairs. Okay? When we say nucleotide base pairs, we're talking about the purines being adenine and guanine, and the pyrimidines being cytosine, thymine, and uracil. Adenine, guanine, cytosine, and thymine being found in DNA, adenine, guanine, cytosine, and uracil being found in RNA. So thymine is for DNA, and it is replaced by uracil in RNA. And the idea is this: your nucleic acids, like your DNA, is a storage. Okay, it stores the code for how to make proteins. Your DNA is basically just a big old honking cookbook for how to make proteins. When you say you've got genes, a gene is a copy of a section of your DNA that codes for a single protein. So when I say you have a lot of different genes, that means you have a lot of different coding regions for protein in your genetic code. Yeah, that'll work. Now, there are two classes of nucleic acids, either DNA and RNA, and this is pretty straightforward stuff. DNA is always found inside the nucleus. It can't escape, whereas RNA is made in the nucleus and can leave and go do work elsewhere. DNA will only be replicated for cell division. RNA will be replicated or made for protein synthesis. DNA is made of adenine, guanine, cytosine, and thymine, whereas RNA is made of adenine, guanine, cytosine, and uracil. DNA is made of deoxyribose. RNA is made of just ribose. And DNA will always be double-stranded. It's in two pieces, strand and strand, whereas RNA is just a single strand, always, because it's a much smaller molecule. And then, of course, there's ATP. Man, look at this, all right? So here is adenine, a pentose sugar, and phosphate, adenine, sugar, phosphate. If we look at ATP, it's adenosine diphosphate. It's an adenine molecule, a sugar, and a phosphate. Man, like these ATP molecules, they look a lot like nucleic acids, so I tend to lump them in there a little bit. Now, let me just make a quick comment. ATP is your energetic molecule. Like all of your tissues are run by ATP. Your cellular metabolic processes are there to make ATP. ATP is going to run your digestive processes. ATP is the reason you're able to stand up. ATP is the reason you can hear me right now. ATP is your cellular energy molecule, and all the energy for ATP is stored in the final phosphate bond. ATP means adenosine triphosphate. Adenosine 1, 2, 3 triphosphate. And what will happen is when you break down ATP to release the energy to use it, what you do is you kick off the last phosphate, and that releases energy kinetically, and you end up with adenosine diphosphate, adenosine 1, 2 phosphates. You can easily rebuild this by resticking the energetic phosphate onto the end of that and making ATP. When it breaks down, it releases a kinetic phosphate, and you're left with adenosine diphosphate. So what your body does is it constantly moves between having ATP and ADP, ATP and ADP, ATP and ADP. It bounces back and forth and back and forth and back and forth and back and forth between these, depending upon its energetic state. So this molecule is not destroyed. It can be reused over and over and over again. And that takes us here to our list of the organic molecules, kind of who's who and what they do. And that's that, man. That's that. So be expecting the quiz on this pretty soon, and your next lecture will be coming up pretty soon as well. Thanks, and have a good day.